Coming up next, we have Dr. Emilee Knowlton. Emilee gained her PhD in infectious disease and microbiology from the University of Pittsburgh under the direction of Professor Charles Ronaldo, identifying immune responses to lytic infection with human herpes virus 8. She joined ProImmune in 2013 after completing her postdoc in Ronaldo's lab. She works in the ProImmune team providing innovative solutions for clients that radically improve our understanding of both desired and unwanted immune responses. She's now a Zoom enthusiast.
All right, well, thank you very much, Andrew, for that introduction. And also thank you, Natasha. It was really nice to hear firsthand how the Pentamers have contributed to your vaccine efforts. And it was a really nice introduction to this talk as well. So I'm going to be providing some examples of how the Pentamers have been used both preclinically and in clinical trials, demonstrating some of their many applications. And then I'm also going to be discussing our class 2 tetramers towards the end of the talk. So one of the major ways to assess the efficacy of vaccines and immunotherapies is to characterize and monitor antigen-specific T cell responses following that vaccination or treatment. So for the cell-mediated response, which you see here on the left, you have presentation of intracellular material. This is typically as a shorter peptide ranging between 8 and 11 amino acids in length. And it is presented by an MHC class 1 molecule, which is comprised of these 3 alpha subunits and a beta 2M. And this MHC peptide complex binds to the T cell receptor on your CD8 T cells. So of course, to detect these antigen-specific CD8 T cells, as we've just heard, we have our Pro5 pentamers, which consist of 1, 2, 3, 4, 5 MHC peptide complexes. Those 5 MHC peptide complexes are configured through a coiled coil domain. And then there are 5 fluorescent tags, which allow for very bright and uniform staining by flow cytometry. These tags can either be APC or PE, or given this unique configuration, we can also provide them with a biotin tag, which gives you the added flexibility in your staining panel in that you can essentially use any color conjugated to streptavidin. Also unique to the pentamers are the ability for us to provide them as unlabeled. And so this has the added advantage of being able to be frozen down and has an extended shelf life. So they're actually guaranteed for a full year. So if you were using these, for example, in your clinical trial, then you can see the advantage in ordering a large bulk amount from a single batch to limit your batch to batch variation. Once you receive them, you can QC them and aliquot them and freeze them down and use them as needed, which would limit the amount of material you actually need to do the QC and gives you that bonus of long-term storage. We have more than 450 different catalog items. You're going to hear about viral hepatitis here shortly, and you just heard about SARS-CoV-2, so you can see we have around 69 specificities on those 2 topics. If we're constantly adding to this catalog, but if we do not have your specific sequence of interest, then we can certainly do a custom reagent as well. And I'm going to be talking a little bit more about that here towards the end of the talk. We have an excellent technical support team that can help to answer your questions. And we also provide some really nice literature in the form of this handbook, which is a very helpful guide that has tools and strategies for experimental planning, the protocols, and even troubleshooting tips. And what sets the pentamers apart from other multimers and really speaks to the consistency of these reagents are the number of scientific publications. There are more than 1,600 references for the pentamer technology. And with that many publications, you can really find examples of several different applications at various stages of research. So I have just a couple examples here. The first is taken out of the University of Pittsburgh, and this is actually a study that I was involved with during my PhD work. And we were using the pentamers in combination with intracellular cytokine staining to look at polyfunctional CD8 T cell responses in individuals with capsule-sarcoma, as Andrew mentioned. This study comes out of the Dana-Farber Cancer Institute, where they use the pentamers to track their cancer-specific T cells following infusion of MART1 CD8 T cells that they had isolated and expanded using their artificial antigen presentation system. And in this example, coming out of University of Minnesota, they actually used the pentamers to stain for allo-specific B cells using immunohistochemistry. So in addition to being very consistent and reliable reagents, they're also quite versatile as well. Now I have quite a few case studies to go through in more detail as well, and a couple that will come out of the University of Oxford. And the first comes out of Ellie Barnes and Paul Kleinerman's group. And here they were developing an HCV vaccine using the adenovirus Ad6 and chimpanzee adenovirus Chad3, which of course is the same technology that's being used for the AstraZeneca Oxford COVID-19 vaccine. And their vaccine strategy focuses on parts of the viruses that are less likely to mutate. And they had selected HCV epitopes that had been characterized from individuals who had spontaneously cleared the infection. And that focused on this A0201 restricted sequence, KLS, as well as this 0101 restricted sequence, ATD. And following the vaccination, they tracked these antigen-specific CD8 T cells using the pentamers. So in part A, you see a representative flow analysis of the A0201 specific pentamer versus CD8. And the upper right quadrant shows pretty high percentages actually of these antigen-specific CD8s at various time points post-vaccination, with the highest being at 4 weeks post-prime and 4 weeks post-boost. And you're looking at close to 6 to 7% of antigen-specific CD8s. And for all of the donors in that study, they did see a consistent range of about 1% of antigen-specific CD8 T cells following vaccination, which was sustained for 36 to 52 weeks. They also included some biomarkers to do some additional characterization on these pentamer-specific CD8s, and that the flow analysis is shown here on the left for those various markers, and then on the right are the cumulative data for those donors. So they were using HLA-DR and CD38 to look at the activation status, which they found was activated early on post-vaccination at week 4, and then remained low for the remainder of the study. They were looking at CD45RA and CCR7 and found that the majority of these cells were effector memory and central memory phenotypes. They also stained for PD1, which is known to be expressed and associated with both activation and exhaustion on persistent virus infection. And they do see a pretty high expression here that then declines over time, which is also consistent with clearing of the viral infection. They also did see expression of CD127, which is associated with a long-term stable memory response. So this paper was quite important as it did show that the vaccine strategy was a success and indicated that the vaccine could work against other HCV strains, and it could possibly remain effective even if some of those targets did mutate. So the next case study comes from the Wakayama Medical University in Japan, where they used the pentamers to monitor the patient's cancer-specific CD8 T cells in a phase 1 trial in response to a peptide-based vaccine. So these patients had advanced or metastatic esophageal carcinoma or squamous cell carcinoma, and they received this peptide vaccination over a 5-week cycle, and it contained epitopes from the novel cancer testis agent, and this was LY6K and TTK. And they saw when they were using the pentamers that there were significant increases in the LY6K specific CD8s here after 12 vaccinations in the upper right quadrant, and then as well as after 16 vaccinations. And this was significantly increased compared to pre-vaccination and against their negative control, which was an HIV pentamer. There were also increases in the TKK specific CD8 T cells as well. So they were able to successfully elicit cancer-specific CD8 T cell responses in these patients, and then identify those using the pentamer technology. Okay, this next case study comes from the Institute Superior de Sanità in Rome, in which they used the pentamers to study EBV-specific CD8 T cells in the immunopathology of multiple sclerosis. So there is evidence that suggests that there is an association between EBV and the pathology observed in MS. So they used EBV-specific pentamers that were restricted to various HLA types of the donors that were used in this study. And these consisted of A0201 restricted, B0702, and B0801 restricted EBV pentamers. And this is an immunohistochemistry stain where they're staining the brain tissue with the 0801 restricted pentamers, EBNA3A, and BZLF1. They did see, of course, positive cells here. And this was sensitive in that they were not able to detect any positive cells when they used the allele mismatch, the A0201. They also used these pentamers in some IFAs and showed using a pooled pentamer approach with the LMP2 and EBNA3A, which were both restricted to O201, that they could detect these antigen-specific CD8s in close contact with a B cell that was infected with EBV. So you see the red are the pentamers and the green are those EBV infected B cells. So they were able to demonstrate a mechanism for entry of those EBV specific CD8 T cells into the MS brain. And that was involved in the CNS tissue damage by mediating an immunopathological response toward a persistent intracerebral EBV infection. And then this last case study also comes out of Oxford, and this is a recent collaboration with our group and Professor Tao Dong's, in which we provided both peptides and pentamers for the characterization of SARS-CoV-2 specific T cells in patients who had recovered from COVID-19. And in this study, they used 28 patients that had mild cases and 14 that had severe cases. And they screened 16 different previously identified SARS epitopes using ELISpot analysis. And they did see responses against 2 NP peptides as well as 1 spike protein peptide. And one of those NP peptides was actually quite strong, this MEV sequence right here, and this is restricted to HLA-B4001. So what you see in this first figure is their flow analysis where they use the pentamer to look at those MEV specific CD8 T cells and to characterize them further. So of course they're gating on their lymphocyte population, singlets, and then gating CD3 and live cells. And then from there they selected for the CD8 positive, pentamer positive CD8 T cells. And within each of the CD8 positive and pentamer positive CD8 populations, they looked at these various markers, including CCR7, CD45RA, CD28, and CD27. And from this, they did determine that a large portion of these cells were central or effector memory phenotype, and they had an early or intermediate activation status. So overall, they did find that there were higher frequencies of M and NP specific CD8 T cells compared to the spike T cells, and that the identification of the T cell specificity and functionality was associated with the milder diseases. And this really highlights the potential importance of including not only the spike proteins, but others as well in future COVID-19 vaccine design. So if you'd like to hear more about this particular data set, we have on our website a link to a presentation given by Professor Tao Dong, who spoke at our Mastering Immunity Symposium in September. So she gives a really nice summary of this data and then also does an interview with my colleague Edmund Neo. So certainly invite you to have a look at those as well. So as I mentioned, we do have both catalog specificities and we do provide custom. Our catalog is quite extensive and we have a range of different antigens available, whether those are cancer antigens or viral antigens, as we've heard, as well as model antigens and autoimmunity. Those catalog items typically ship out in 1 to 2 days to 1 to 2 weeks, just depending on that specific sequence. And again, if we don't have your sequence of interest, we can make a custom at no additional charge. So we've been providing the pentamers for quite a long time, and during these years, we've improved our manufacturing efficiencies. And so I'm very happy to say that this year we have dropped our prices pretty considerably, and there's no difference in cost between a catalog and custom reagent, and there's no longer a custom setup charge associated with the custom reagents. So if you would like a custom pentamer, it's a very straightforward process. It's about a 3-step process. You get in touch with one of the ProImmune representatives and provide the allele of interest as well as the sequence. And we would evaluate that sequence to look at the anchor residues to determine whether or not it may make a stable pentamer or tetramer, depending on if you're also working on class 2. And then once accepted, we would initiate that synthesis. And that does include synthesis of the peptides, so you don't actually have to provide us with any material at all. And then it's about a 4 to 6 week process until we then QC them and ship them out to you. And what we have available in terms of alleles are shown here in this table. We have 16 different human alleles, 5 different mouse. This covers Balb C and Black 6 mice, 2 Mamu sequences, and then also a chimeric O201 KB as well. Okay, so now to switch gears a little bit toward class 2, of course, to look at the antibody-mediated immune response or the antigen-specific CD4 T cells, we have a very similar technology. In this pathway, there's presentation of extracellular material, and the peptides here are typically a bit longer, generally around 15 amino acids in length, and they are presented by an MHC class 2 molecule, which is comprised of 2 alpha and 2 beta subunits. That MHC peptide complex then binds, of course, to the TCR on the CD4 T cells, and for that we have our class 2 tetramers, which have 4 MHC peptide complexes. Now these, like the pentamers, are, they're ready-made, they're labeled. You can get them with either APC or PE. They have a couple different quantities, 50 tests and 150 tests. And we have 132 different catalog specificities. And again, you're going to be hearing about viral hepatitis in just a moment. So you see we have around 15 for that specificity. They do also ship out in 1 to 2 days or a couple of weeks, just depending on that sequence. And we also provide custom, which has a similar synthesis program as the pentamers. We have 9 different class 2 alleles available for human and 3 for mice. And these also take about 6 weeks or so. They are guaranteed for 6 weeks. And if you do want to get them as monomers, we have biotinylated monomers as well, and you can do that tetramerization yourself as a part of the kit. So I have just a couple of quick examples of staining here. So this is also coming out of Ellie Barnes' group. So we're looking here at these HCV vaccinated donors. The upper panel shows on the y-axis your tetramer. There's both a DR2. This is the same DR2 sequence, and the CD4 is your x-axis. And this is at 12 days culture and at 22 days culture. So you see very nice staining populations at each of those time points. And again, this is quite specific in that you don't see any positive CD4 T cells when an allele mismatch is used. And this is a nice segue into our next talk when we hear from Leah from Mass Gen. This is an example of staining of 2 different tetramers, a DR4 specific and a DR1 specific tetramer versus CD4. And you get nice distinct populations in the 0.07 range as well. So we look forward to hearing Leah's talk in just a moment. I did just want to give a little plug for our Mastering Immunity symposium that will be here just in a few weeks on March 25th. We had a very successful conference back in September when we did the virtual event, and a lot of really nice presentations came out of that. So I certainly invite you to join here in a few weeks, and the list of the speakers is shown below. And you can also see the link to find out more information about this conference. So today I've gone through the pentamers and tetramer technology, which is part of our immune monitoring reagents. We certainly also offer immune monitoring services as well, and that's a large part of what we do, including ELISpot analysis as well as intercellular cytokine staining. We can also provide HLA typing services so that you can identify the HLA restrictions of your donors so you can use these reagents. And on the other kind of arm of what we do are immunogenicity assessment tools. So you see a very large list of immunogenicity assessment assays as well. So myself or Andrew would be happy to talk to you about any of these if you have interest in doing so. So here in summary, our class 1 and class 2 multimers are very powerful and widely used immune monitoring tools that allow for characterization of antigen-specific CD8 and CD4 T cells, ultimately helping to determine potential correlations between treatment and clinical outcomes. And this is a very important step in a successful epitope discovery and immune monitoring program. So not only can we offer these tools to monitor the responses yourself in-house, but we can offer a range of specialist assays to support your research as well. So we are the world expert and leader in MHC multimer technology field, and our experience in this area will, with our integrated platform, will not only save you time and money, but also help to reduce your overall project risk. So with that, I would be happy to answer any questions. Thanks for listening.
Thank you, Emily.